Right, so here's Thomas Young. He has a good idea. He gets a bad referee report. You got to nail the case. And he didn't actually do that. It took a little while to do it, but you've got to find a way to nail the case. And what you need, physicists are the people who believe that to nail the case, you need quantitative, detailed predictions from your theory. If your theory predicts 100 numbers, and you go to the lab and you make your measurements, and those numbers come out correctly within their appropriate error bars, it's probably not an accident. Probably means that theory was promising. Lots of theories fail right there at that point and get tossed into the trash. So that's what physicists like to do. It's our discipline. Don't go too far in a tangent without some authority from experiment. Ideally, we'd like a whole lot more experimental data points than we have unknown parameters. How do you do it in this case? Well, here's a classic experiment that was done by many people. You take a kind of light, maybe it's magenta, some light that I'm going to call the target light. The human subject is looking at it projected onto half the screen. Then there are these three standard lights. Maybe they're red, green, and blue, and maybe there's some other choice, but there are three standard lights, and the subject has got these three knobs. And you say to the subject, okay, dial those knobs, please, until you get a perfect match, and you can't tell the difference between the two halves of the screen, if it can be done. Okay? That generates a lot of data. That generates data that look like this. Here are a bunch of different target lights, all sorts of different target lights. You ask the subject to do that task, and for each one, the subject coughs out three numbers: how much red, how much green, how much blue. That's what these three curves are. That's the quantity of red, green, and blue light needed to match that light. If you can make a theory that predicts all that, it might be a promising theory, don't you think? That's a lot of quantitative data. How does it work? Well, once you've measured those sensitivity curves, here they are for humans and other primates. We've got red and green, and yes, they do overlap quite a lot, and there's blue off to the side. There's an interesting reason for that. I don't want to get into it. But those are the sensitivity curves, and once you've measured them, you can predict the response of each of those three photoreceptor cells to any kind of spectrum of incoming light. Just color by color, you multiply how much of that color there is, times the sensitivity at that color, you add them all up, and that's your answer. Okay? You do that to the target light, you get three numbers. Then you do that to the three standard lights, and you get numbers. And then you say what combination of those standard lights would needed to mimic the response elicited by the target light. Okay? Mathematically, it boils down to solving three linear equations. And I'm allowed to say that to you because I just recently discovered that high school students now do that in high school, solve three linear equations and three unknowns. So what happens? Well, here's that graph before, but I didn't tell you everything about that graph. These solid curves are the experimental data that came off the experiment. These dots are the predictions of theory with no free adjustable parameters at all. Once you measure those sensitivity curves, the die is cast, there's no wiggle room, there's no fudge factors, and it comes out on the money. It's very strong evidence for that theory. That's what scientists do all day long, is try to get something that good. Costs a lot of effort. Okay, now we're ready for some technological payoffs. We've done our homework. You can fool the eye into thinking that a whole range of colors is present. If you take a look at an old-fashioned television set with a magnifying glass while it's on, you see it doesn't have millions of colors. It has three colors. It's got red, green, and blue glowing things. And just by adjusting the relative proportions of that, the television is able to mimic all the millions of colors that we like to see. And similarly with computer screens. So that's cool. That's technology. It's not very recent technology. But let's turn it around. What this means, the fact that you can match a lot of colors with just three means that our eyes are discarding a lot of information about the spectrum of light entering any given visual field. And we could ask ourselves, could some artificial visual system do better than that? Now that you know about our eyes, it opens your eyes, so to speak, to something you could do better. You know, if you're going to have a baby, you might get genetic counseling. You might get genetic testing. And what do? They don't sequence the whole genome. No, only Bill Gates gets that, you know. But there's an old test that reveals a lot of problems called karyotyping. You take cells and you stain the chromosomes at the right point in the division cycle, and you used to snip them out with scissors and line them up so you see they come in pairs, right? And here's one chromosome and its partner, and here's another one and its partner, and you look at them, and if you see that, well, there's three copies of this chromosome and there's only supposed to be two, that's a bad sign. If there's only one and there's supposed to be two, that's a bad sign. If one of these has a whole huge chunk missing, that's a bad sign. It's kind of a blunt instrument, isn't it? It's useful. It lets a lot of things. It catches a lot of things. But wouldn't it be nice if you could take each one of those chromosomes, take chromosome number 1 and paint it all one color, chromosome number 2, paint it all some other color? Maybe you'd learn more like that. So this is not a talk about molecular biology. So all I'm going to say is that smart people like my friend Yuval Garini know how to do that. And great, he's labeled all the chromosomes, each with its own color. Doesn't tell you much, does it? The colors that are available in fluorescent dyes are not really all that distinguishable to our eyes. You look at this, and if there's more than, say, 2 colors, it's hard to say what's going on. But maybe we could do better than that. Suppose instead of 3 sensitivity functions, you tested that each pixel of the image with 10 or 100. If you have these narrow sensitivity functions, you could tell the difference between yellow and green plus red, because yellow would excite only this one, and green plus red would excite only those 2, and they're different, and you could tell the difference. You'd get better color discrimination. Well, there's a gizmo that does that. It's a Sagnac interferometer. I wish I had time to tell you about how it works, but effectively what it does is it takes every pixel in the image, sends it through a prism, spreads it out into its spectrum, sees how much of each color is present, and turns that into a graph. Okay? That's what the interferometer does. And it's a gizmo that was invented. Physicists think of this particular gizmo as something that was used to confirm some predictions of Einstein's theory of general relativity, not for this purpose. Other people think about it as useful in the guidance of ballistic missiles. A good spy has a whole catalog of all the gadgets out there when some new nugget of intelligence needs to be found. What's it do for you? Here are 2 popular fluorescent dyes. One's called Psi-3 and one's called Texas Red. The green line is the spectrum of Psi-3. The blue line is the spectrum of Texas Red. They're similar, but they're not quite the same. These peaks are a little bit off. Those peaks have a different height. But if you just look at it with your eye, you're going to see red. There's 600 nanometers, and this you can't see because it's infrared. You look at this with your eye, you can't tell the difference. There's a third curve here. That's what happens when you have both of them present. It's the sum of those 2 spectra. If you label all of chromosome 7 with Psi-3 and all of chromosome 13 with Texas Red and all of chromosome 4 with a combination, you get a picture that looks like this, and once again, not very interesting, not very useful, hasn't told us much. But you can do better than that. Each pixel on this picture, you can send it through that interferometer and make a digital decision, which of these 3 curves does it most resemble? You know it's got to be one of those 3 because that's what you labeled it with. Okay, that digital decision can be rather precise, and then once you've made your decision, you can change that color into some fake color that humans are good at discriminating and get a more informative picture. Okay, the doctor can see you now. Okay, these guys are a pair, those guys are a pair. Now you can see what's going on. That's the spectral karyotyping idea. That's what I mean by superhuman color vision. Let's go back to this. This is an actual karyotype from a guy whose son was born with mental retardation. That's a bad thing. Chromosomes look pretty good until you look at the spectral karyotype, when you see a whole big chunk of one chromosome has been swapped with another. Everything that's supposed to be there is there, but in the wrong place. There's some gross abnormality there that ordinary karyotyping was powerless to reveal. I think that's pretty good. Here's another one. This one looks all scrambled up. This is the karyotype from a cancer cell. Okay? We're entering a period of personalized medicine. Different strains of cancer cells have different karyotypes. They have different genetic sequences. Someday there's going to be an app for your doctor's iPad that says this strain is sensitive to that drug and not sensitive to that drug. And this is one of those techniques, one of many, that are going to start giving us personalized medicine. Here's one I like a lot. This is a karyotype from my cousin. But don't worry, he's okay. He's your cousin too. This is a primate. It's a gibbon. Okay? And when people say to you, oh yeah, those primates, they've got 99% the same DNA as we do, it's true as far as it goes. Look, every label that sticks to our chromosomes also sticks somewhere to his chromosomes. But look at all that shuffling that's been going on. This picture is full of clues about our evolutionary lineage. It's pretty interesting. It tells you something you couldn't see without superhuman color vision. Here's another example. If you take a slice of brain tissue and you just stick it under the microscope and look at it, it's a mess. There's this tangle, this dense tangle of all these things tucked cheek by jowl. They're long, skinny things. You can't see anything. You can't discriminate anything. Wouldn't it be cool if you could take every neuron in a slice of brain, give it its own different color, stain it uniformly all up and down its whole length, its personal color, which is different from the colors of its neighbors? Then you could see a lot more. That's the rainbow method. It works essentially by the same mechanism I've just told you about. Now you can see what's going on in there. You can also see it in 3 dimensions. That's pretty nice. And you can also see who's connected to who and how. Here's a neuron. And you see its output is connected to this other neuron's input, and it winds around. You get all sorts of fantastic detail. There's a whole new branch of science called connectomics that didn't exist until methods such as this. This is not the only method. Started coming online in just the past few years. Now we're going to be able to see things we couldn't ever see before because we have superhuman color vision. So that's it for part one. If we didn't get that understanding of our own vision with its failings, we might never have imagined the possibility of doing better, nor the means to do so. That's what I mean when I say spycraft. You know, when people say truth is beauty and beauty is truth, you know, I kind of glaze over because usually they're just blowing smoke at you. But I couldn't help feeling when I looked at those rainbow pictures that I'd seen it somewhere before. So I'm lying on my back getting my root canal a couple of days ago, and suddenly it popped into my head. I got this vision. Oh yes, it's this painting by Ted Geisel. Doesn't it look familiar? You may know him by his pen name. So I do believe, though, that great art can reveal to us truths that are not visible yet to empirical science. And in that connection, I'd just like to draw your attention to that little worm there. Empirical science hasn't seen that yet. This, you know, Professor Pagels, this may be more in your line than in my line, but just remember who you heard that from first, okay?